Okay, we are continuing with our chapter of microorganism and their use in biotechnology. In our previous lecture, we saw the basic structural differences between fungus, virus, and bacteria. We saw their comparison between the sizes and the structure. In today's lecture, we will see the differences between their mode of nutrition and reproduction. If we see the mode of nutrition in viruses, we see that they do not feed themselves. They require to have a host cell for reproduction and for their nutrition as well. Viruses can feed on plant cell or animal cell and or on even bacterial cell. If you remember that bacteriophages were the viruses that were feeding on bacterial cell. On the other hand, bacteria can be autotrophic, that they can make their own food like plants. Then they can be parasitic, that they need to have a host cell to feed on them. Then we also see that bacteria are saprophytic in nature as well, because they are feeding on the dead decaying matter of plants and animals. This makes them decomposers. Fungus can be parasitic, that it can feed itself by attaching itself to the host cell. They can be saprophytic, just like bacteria, that they feed on dead decaying matter. So if we see that the decomposers in the world are majorly bacteria and fungus. In mode of replication, you will see that the viruses take controls of the host cell machinery. In the previous lecture, I showed you a picture or a animation where the viruses were attaching itself to the bacterial cell, controlling its cell machinery and asking it to produce the components of viral structure so that more viruses can be produced inside the host cell. Bacteria, on the other hand, reproduce by asexual reproduction, which is binary fission. Fungus produce by budding. This budding is seen in yeast. And in some multicellular fungus, we see that the reproduction is via spore production. The reproduction in bacteria, like in the previous slide I told you, is by binary fission or asexual reproduction. What happens here is that the parent cell divides into two simply. The offspring which is formed is identical copy of its parent. It's a clone of its parent. Nothing changes genetically in the daughter cell. First, the genetic material divides and the cell divides later, giving rise to another whole new cell which is genetically identical to the parent cell. So this is a very simple process of reproduction. The rate of reproduction is quite fast. If you see here, if you take one bacterial cell and provide it with all the nutrients it requires to reproduce, this one bacterial cell will give rise to millions and trillions of the cells. Each would be the copy, identical copy to its parent, and you can get a colony of bacteria in no time. Reproduction in fungus is by budding. In yeast, in unicellular yeast, we see that a bud is formed, a little cell is formed from one side of the parent cell, and when it reaches to the approximately the same size of its parent cell, it separates itself from the parent cell. So this is budding, where a small bud forms first and then it grows in size, and once it reaches its maximum size, it deattaches itself from the parent cell. On the other hand, fungus reproduce by spore formation, which is seen in bread mold and rhizopus. I told you that the vertical hyphae contain spore producing bodies called sporangium. They produce many spores which are dispersed into the air when the sporangium bursts, when it gets matured. The spores, on landing on a suitable substrate, provided that they get suitable pH, temperature and moisture, would lead to formation of a new mycelium.